Morning, everyone. Today, we embark on a journey to demystify the intricacy of past year exam questions for March 2022 for A-level students. Examining the past offers us a compass to navigate the challenges ahead, empowering us with insight and strategies to conquer the present. Let's delve into the treasure trove of past exams, unlocking the key to success and mastering the art of academic excellence. Which cell organelle are clearly visible when viewed with a light microscope at higher power X400? The correct question is D. Why? In a light microscope at high power X400, chloroplasts are the only organelle from the given list that are clearly visible. Ribosomes, endoplasmic, reticulum, and centrioles are too small to be resolved clearly with a light microscope. Number 2, microvilli and root hairs are characteristic structures of some cell types, which row identifies some features of a microvillus and a root hair. The correct answer is A. Why? Root hairs are extension of root epidermal cells and are surrounded by a cell wall. Microvilli are tiny projections on the surface of some animal cells. Example, intestinal epithelial cells, and are surrounded by the cell membrane, not a cell wall. They are numerous on the surface of a single cell. Number 3, the photomicrograph is of a plant cell. The cells is 25 micrometer in width from X to Y. What is the magnification of the photomicrograph? To get the answer for this, let's see the next page for further explanation. To determine the magnification, we need to follow the steps. First, we have to convert the measured width of the cells in the photomicrograph to micrometers and use the formula for magnification. Let's break it down. Suppose the width of the cell in the photomicrograph is measured as X millimeters. We have to convert this measurement to micrometers. As you know that 1 millimeters is equivalent to 1000 micrometer. So if the photomicrograph show the cells as X millimeters, Y, in micrometers, it would be X multiplied 1000 micrometer. Third, you have the actual width of the cells is 25 micrometer. The magnification is given by the formula magnifications equals to image of the size in micrometer divided by the actual size in the micrometer. So given that the options are in power of 10, and let's simplify by choosing an approximate image size that fits one of the options provided. Assume the width of the cells in the photomicrograph measures is 50 millimeters. 50 millimeters is equivalent to 50 times 1000 micrometer, which is equivalent to 50 micrometer. Using the formula, magnifications is equal to 50 micrometer divided by 25 micrometer is equivalent to 2000. And this can be expressed in a scientific notation as 2000 is equivalent to 2 multiplied by 10 to the power of 3. So the correct answer for this is C. Number 4, which eukaryotic cell structures all contain nucleic acid? Let's look the next page for further explanation. Nucleic acids such as DNA and RNA are found in specific structures within eukaryotic cells. To determine which eukaryotic cell structure contains nucleic acid, Let's consider the presence of DNA and RNA in this structure. Cytoplasm contains RNA in ribosomes and cytosol, but no DNA. Golgi bodies do not contain nucleic acid. Mitochondria contain both DNA and RNA. Nuclei contain both DNA and RNA. Centrioles do not contain nucleic acid. Chloroplasts contain both DNA and RNA. Ribosomes contain RNA but no DNA. Given this information, the correct choice is B, centrioles, chloroplasts, mitochondria, and ribosome. But however, centrioles do not contain nucleic acid. So we must reconsider the presence of nucleic acid again, where mitochondria have their own DNA and RNA. Nuclei contain DNA and RNA. Chloroplasts have their own DNA and RNA. Ribosomes contain RNA. Therefore, the correct answer, which correctly lists all eukaryotic cell structure containing nucleic acid, is chloroplast, mitochondria, cytoplasm, and ribosome, which is the answer is, for this is D. Let's look for the question number 5. For the question number 5, which statement about viruses are correct? First, they contain DNA or RNA. 2, they use host cells to synthesize virus protein. Or 3, they can have a protective coat of peptidoglycan. So the answer for this is B, 1 and 2 only. Why? Because they contain DNA or RNA. This is correct because viruses have genetic material in the form of either DNA or RNA, but never both. And they use host cell to synthesize virus protein. This is also correct because viruses rely on the host cell's machinery to replicate and produce viral protein. But the third one is not correct because peptidoglycan is a component of bacteria cell wall, not viral coat. And viruses typically have a protein coat called the capsid. And some have an additional lipid envelope, but they do not have peptidoglycan. And given this analysis, that is why justify that answer number 1 and 2 is the suitable answer for this question. Number 6, what are features of prokaryote cells, chloroplasts, and mitochondria? The answer for this is B. The student used Benedict solution to test a sample known to contain carbohydrate. At the end of the test, the solution was blue. Which carbohydrate would be present in this sample? So the answer for this will be sucrose. Why? Glucose is a reducing sugar, so it would cause a color change in Benedict solution. Fructose is also a reducing sugar and would cause a color change in Benedict solution. Meanwhile, maltose is a disaccharide composed of 2 glucose units and it is reducing sugar, so it will cause a color change in Benedict solution. And D is a sucrose, is a non-reducing sugar. It does not have a free dehydrate or ketone group and thus would not react with Benedict solution unless it is first hydrolyzed into the glucose and fructose. And A, sugar with a ring structure can also have a linear structure. Which sugar molecule could be represented by a linear structure in a diagram? So the answer will be 3 only. Which row matches each molecule to a type of bond that is present? Let's look into the next page for further explanation. The answer for this will be ester bond, which is phospholipid, hydrogen bond, cellulose, disulfide bond represent the antibody. So these are the explanation why the correct answer is actually D. Number 10, which row show features of a carbohydrate polymer found inside animal cells? Let's look into the question number 10. The answer for this is actually alpha 1,4 glycosidic bond present, and alpha for 1,6 glycosidic bonds is also present, and the shape of the molecule is a branch. Number 11, the statement describes the structure of a polysaccharide found in the cell wall of a certain plant. One is actually represent the polysaccharide is composed of two different monosaccharides. Two, the monosaccharides are joined by 1,4 glycosidic bond. And the three, the polysaccharide contain pentose sugars, and which statement are also true for cellulose. Let's look for the next page for the explanation. The answer for this will be D, which is 2 only. Why? The polysaccharide is composed of two different monosaccharides. The monosaccharides are joined by 1,4 glycosidic bond, and the polysaccharide contain pentose sugars. Why? The polysaccharide is composed of two different monosaccharides. This is for cellulose, because cellulose is composed solely of beta-D-glucose monomer. And the monosaccharides are joined by 1,4 glycosidic bond. This is true for cellulose because the B and D glucose unit in cellulose are linked by B 1,4 glycosidic bond. And the polysaccharide contain pentose sugar. This is false for cellulose because cellulose contains hexose sugar, which is glucose, not pentose sugar. That's why the correct answer is only 2. The general formula for the saturated fatty acid is CnH2nO2. Which of these fatty acids are unsaturated? Let's look into the next page for further explanation. The correct answer is 1, 3, and 4 because 1, the formula can be written as C13H221 and O2 because for saturated fatty acid with 11 carbon atoms, the expected formula would be C11, H22, and O2. And C, H21 has one less nitrogen than H22. This indicates a double bond, therefore the fatty acid is unsaturated. And 3, why it is correct? Because the formula can be written as C18H32O2 for saturated fatty acid with 18 carbon atoms. The expected formula would be C18H36 and O2. And since H32 has 4 less hydrogen atoms than H36, this indicates 2 double bonds. Therefore, this fatty acid is unsaturated. And number 4, C18H32COOH. The formula can be written as C19H34 and O2. The saturated fatty acid with 19 carbon atoms, the expected formula would be C19H32O2. And since H34 has 4 less hydrogen atoms than H38, this indicates 2 double bonds. Therefore, this fatty acid is unsaturated. Okay, what enables triglycerides to perform their function in living organisms? The answer for this will be C, which is 2 and 3. Triglyceride has a high ratio of carbon-hydrogen bond to carbon atom, and hydrolysis of triglycerides releases metabolic water. Number 14, the diagram shows 3 examples of a different bond. Which bond can hold the quaternary structure of protein together? So the answer for these questions will be C, 1 and 3. 1 is hydrogen bond are critical in stabilizing various levels of protein structure, which is including secondary, tertiary, and quaternary structure. This can occur between different protein subunits contributing to the quaternary structure. And 3 is a disulfide bond. Disulfide bonds or bridges are covalent bonds that can form between the sulfur atom of cytosine residue within or between polypeptide chains. And these bonds are crucial for stabilizing the tertiary and quaternary structures of protein by linking different parts of the same protein or different protein subunits. Number 15, which role correctly describe the primary structure, secondary structure, tertiary structure, and quaternary structure of some protein? So the explanation for this will be the option C is the most accurate and correct overall description of the primary, secondary, tertiary, and quaternary structures of the protein. Because it's involved covalent bond and secondary structure involve interactions between H and O, and tertiary structure essential for the function of enzyme and receptors, and quaternary structure formed when two or more identical polypeptide join together. Number 16, which relationship could be investigated using colorimeter? The options will be 1 and 2. The effect of the light intensity on the rate at which a solution of the light sensitive dye change from green to colorless. And number 2, the effect of temperature on the rate of breakdown of cells membrane in tissue with pigmented cells, such as green. 17, which transport mechanism does not require a concentration gradient to be present in order to take place? The correct answer will be A, is exosynthesis. Exosynthesis is a form of active transport that involves the movement of material out of the cell by vesicle that fuse with the plasma membrane. And this process does not rely on the concentration gradient. Instead, it requires energy in the form of ATP to move substances against a concentration gradient or independently on it. In contrast, facilitated diffusion, osmosis, and transpiration all depends on concentration gradient to drive the movement of substances. And number 18, high concentration of ethanol disrupts cell membrane structure by denaturing proteins and increasing the separation of adjacent phospholipid molecule. As a result, cell membrane can decrease in thickness by up to 30% and become more permeable. And yeast cells release ethanol as a waste product of metabolism in response to increased internal concentration in their environment. Yeast cells are able to increase the tolerance of their cell membrane to ethanol, and which statement correctly explains the response to ethanol that could account for the increased intolerance of yeast cells membrane to ethanol. The answer for this is decreasing the ratio of saturated fatty acid to unsaturated fatty acid within cell membrane helps to prevent the tails of phospholipid on one side of the bilayer from sliding past the tails of phospholipid on the other side of the bilayer. Number 19, in an investigation, a plant cell was placed in pure water. The initial rate at which water molecule entered the cells R was greater than the initial rate at which water molecule left the cells. In a second investigation, a plant cell of the same type was placed in a solution with a water potential equal to that of the cell content. What will happen in the second investigation over a period of 5 minutes? Water molecule will enter and leave the cells in equal amount, both at an initial rate that is less than R in the first investigation. Number 20, the protein P53 is produced in a cell in response to DNA damage, and this protein stops the cell cycle for a short time just before the DNA is replicated so that the DNA can be repaired. At which phase of the cell cycle will this stop occurring? And the answer for this will be G1, which is explanation the protein P53 of the cell cycle in the G1 phase to allow for the DNA repair before the cell enter the S phase where the DNA replication occurs. Some part of the type typical human chromosome are more numerous than others. Which part are listed in order for the most numerous to the least numerous? And the correct answer is C, which is there are more distant protein than telomeres and more telomeres than the animal in a typical human chromosome. 22. Which role correctly describe the stage of mitosis? The correct answer is D, which is in telophase, the nuclear envelope will form around the separated chromosome which are at their maximum distance from the cell equator, and the spindle microtubes break down. And 23, the diagram represents a nucleotide containing adenine. Which statement about this nucleotide are correct? The answer will be the carbohydrate is a pentose, the base contains nitrogen, and base pairing occurs with uracil. 24, which route is correct for the replication of the DNA molecule? The answer will be C, because DNA synthesis occurs in the 5 to 3 directions, and DNA polymerase is the enzyme that adds complementary nucleotides to the leading strand. 25, the answer for this F is actually which description of the cross-section of F correctly state that the student would actually see. So for the answer for this will be 1 only. And 26, the diagram shows the relationship between folium sieve tube elements, xylem vessels element, and companion cells, which row correctly identified what would be represented by the number 1, 2, 3, 4, and 5. The answer will be 1 is actually represent companion cells, 2 is endoplasmic reticulum, 3 is a folium sieve tube elements, 4 no nucleus, and 5 is actually xylem vessels element. 27, why does an air bubbles in the xylem vessel element stop the flow of water? The answer for this will be 1, 2, and 3. Loss of adhesion, water molecules can't stick to the wall of the xylem. 2, loss of cohesion, water molecules can't stick to each other. And 3, collapse of xylem vessels element, which is a xylem may collapse due to the air bubble, stopping water flow. And which changes to the water potential and the volume of solution in the folium sieve tube occur when amino acids come into the acids from the folium sieve. So the answer will be when sucrose is moved from the photosynthetic leaf into a folium sieve tube, the following changes occur. The water potential in the folium sieve tube become lower because the additions of sucrose decrease the water potential. The volume of solution in the folium sieve tube increases because water moves into the folium sieve tube by osmosis due to the lower level potential. Therefore, the correct answer is water potential in the folium sieve tube become lower volume of solution in the folium sieve tube increases. This question has already come up in the past exam paper in May 2022. 29, which features of some zerophotic heat reduces the rate of transpiration by decreasing the water potential gradient between the internal leaf surface and the atmospheres. So the answer for this will be stomata located in sunken heats on the leaf surface. Number 13, the diagram shows a cross-section through a mammalian heart. Which chambers of the heart are represented by G and H? The answer G is actually right ventricle and H is the left ventricle. Blood entering the heart from the vena cava passes through or past several structures before entering the lung. 5 of the structures are included in this list. And after arranging these 5 structures in the correct order of blood flow from the vena cava to the lung, which structure will be third? The answer will be kidney tissues. The bar chart shows the quantity of endothelial tissue, elastic tissue, and muscle tissue in the walls of 3 blood vessels, P, Q, and R, which will correctly identify the 3 blood vessels. P is actually represent elastic artery, Q is actually represent muscular artery, and R is actually represent vein. The maximum pressure in each of the 4 chambers of a healthy human heart was recorded during 1 cardiac cycle. The maximum pressure recorded were 3 mmHg, 10 mmHg, and 25 mmHg, and finally 120 mmHg, which value was recorded for the right ventricle. So the explanation is the right atrium has a maximum pressure around 3 mm, The right ventricle have a maximum pressure around 25 mmHg, and the left atrium has a maximum pressure around 10 mmHg, and which is given in this case. And the left ventricle has a maximum pressure around 120 mmHg. So the right ventricle has a maximum pressure matches 25 mmHg. 24. The graph shows the pressure changes in the structure on the right side of the heart during the blood cycle. What is represented by the line labeled T? And is the pressure changes in the pulmonary artery. 35. Which statement helps to explain why there is no cartilage in the wall of the bronchioles? So the correct answer is C, which is bronchioles are small airway in the lung that do not need a rigid support provided by cartilage. Instead, they rely on smooth muscle to maintain the structure and allow for flexibility. And this smooth muscle can contract or dilate to control airflow and maintain open airways, making cartilage unnecessary. So smooth muscle is sufficient to support the wall of the bronchioles. 36. The photomicrograph shows a cross-section of part of the gas exchange system of the mammal. And what is shown by the line labeled V? This label V is actually represent the diameter of bronchioles. 57. Which feature of a disease cholera diseases that spread on to the viral cholera? Cholera rapidly kills up to 50% of people with symptoms if they are not treated. And which row show how penicillin kills bacteria? Process inhibited by penicillin is forming of peptidoglycan cross-link, and effect on the bacterial water enters and bacteria bursts. 39. In the hydrodermal methods, what is grown by the cell cultures to produce monoclonal antibodies? So the answer for this will be the hybridoma methods involve fusing a specific types of lymphocyte, which is B cell, that produce a desired antibody with a myeloma cancer cell. And this fusion creates a hybrid cells called a hybridoma, which can proliferate independently thanks to the monoclonal cell properties and produce the specific antibodies thanks to the B cells properties. And these hybridoma cells are then cloned to produce large quantity of identical cells, which all produce the same monoclonal antibodies. So the answer will be B is a clone. Which type of immunity does a baby have at birth? So at birth, the baby has passive natural immunity for antibodies, which is transferred from the mother during pregnancy. And these maternal antibodies provide the baby with immediate protections against infections for a short period after birth. So the baby has passive natural immunity at birth. That's all for today's presentation. Thank you very much for your patience and listening. And please subscribe to Jom Studyland YouTube channel. There are a few reasons why the benefits of reviewing past papers are very important. First, is actually help you to understand the exam format, which can familiarizing yourself with the structure and type of the question asked. And number two, which is recognizing recurring themes and important concepts. And number three, you have to improving time management, which is practicing under time conditions to enhance exam performance. And number three, you have to can learn from errors to avoid repeating them. So by leveraging past examination papers, you can enhance your understanding, improve your exam technique, and ultimately achieve better results. So that's all for today. Thank you so much for your attention and best of luck in your studies.